Hello, my name is Jada Zaragoza,
And my name is Rothmila Tajrian.
and we did our research project on antimicrobial resistance in bacteria isolated from meat products, specifically on ground beef.
So you might be wondering, what is antimicrobial resistance? Antimicrobial resistance is basically us humans. We tend to get sick quite often in our lifetime, along with animals. They also get sick quite often in their lifetime. Whenever we get sick, we are always prescribed antibiotics. Along with the animals, they also get prescribed to take antibiotics. And we take antibiotics not just once, but quite often within our lifetime. And once them so frequently, we tend to have bacteria that mutate against the antibiotics that we are taking in, and they tend to not work with our body and work against the antibiotics and resist against the antibiotics, which is not a good thing to have within our body because we would want these antibiotics to help us get rid of this viral infection within our immune system. Along with animals, they also build up that resistance, which we tend to consume these animals and consume the resistance within them goes into us because through the nutrients that comes from the more so we're focusing our research on ground beef. So the ground beef we're consuming is has antibiotic resistance and we are consuming that antibiotic resistance, and that is not a good sign to have in our body because we would want these antibiotics to work with us. So our research question is, can antibiotic resistant bacteria be isolated from ground beef products? What antimicrobial agents are these bacteria resistant to? And within this research, a couple of our materials and methods, we did serial dilutions, we plated and cultured our bacteria, we did some tea streaking, biochemical testing, gram staining, and along with that, we also did antibiotic resistant testing as well.
So what we did was we grabbed approximately 1 gram of meat from 3 different locations. We put them in 50 milliliter conicals and we labeled them A, B, and C. And we also put 10 milliliters of PBS solution in these conicals. And the reason why we used PBS solution was to keep the cells in an isotomic state so they don't shrink or they don't swell. We vortexed them to create a slurry solution. And we basically did a bunch of serial dilutions on 3 different types of media: nutrient agar, EMB plates, and McConaughey plates. And the reason why we used 3 different types of plates was because we wanted to get as much growth as possible. The reason why we did it on nutrient agar was because nutrient agar isn't selective or differentiative at all. With the McConaughey plate, it selects for gram-negative and for lactose-fermenting bacteria. With the EMB plate, it selects for gram-negative as well and pathogenic bacteria. So once we got our serial dilutions, we tried to look for a bacteria that was dark in color, a similar appearance to E. coli. And then we did tea streaking, and we did find E. coli, and we knew that it was E. coli because of its green metallic color that it left on the EMB plates. That came from grocery store B. In grocery store A and C, we were unsure exactly what type of bacteria they were. In the middle, you can see grocery stores A bacteria. It was like a light purplish color. Grocery store C, its colonies were orange in color. And since we didn't know what type of bacteria they were, we did a bunch of biochemical testing for them. So on the first picture of the tubes, you see we did sim tube testing, and that basically tests for motility. Both bacteria did show signs of motility. Then we did testing for citrate and urease, and they were both negative for urease. Then we did gram staining, and we knew that both these bacteria were gram negative, but we wanted to see the shape the bacteria were, and they both were broad shaped. Then we did oxidized production testing on the paper strips, and they were negative for that since no purple colored had appeared. Then we did catalyst production testing, and they were both positive for that, as you can see, like the tiny little air bubbles that appeared once we added the hydrogen peroxide. And then we tested fermentation of sugars. They both did not ferment lactose whatsoever. They were, as you can see, the color didn't change to like a light yellowish color, but they did both ferment glucose and arbanose. And then we did tested for antimicrobial resistance.
So with our antimicrobial resistant testing, the left two columns of the plates were our A10 to the -2. The middle ones were from market B, and the one on the very right is from market C. And we did 6 plates for each isolate, 3 different trials to make sure that we were very accurate with our results. And we noticed that market store A had resistance against penicillin, erythromycin, and ampicillin, and market B had resistance against erythromycin only, versus market C had no resistance whatsoever.
So after we got done with our biochemical testing, we found that the bacteria that came from grocery store A was E. aerogenes, and then the bacteria that came from grocery store C was P. aeruginosa. So all these types of bacteria that we ended up finding are completely normal to find in ground beef, except they're all pathogenic, and it's the reason why you're supposed to cook your meat. We we did see that they did have varying profiles of antimicrobial resistance, which is not normal whatsoever. So the findings from this project basically just showed us that we need to be careful what we eat, and we need to monitor antimicrobial resistance in foodborne pathogens.
Thank you for listening to our research.
Thank you so much.